The goal of this project was to begin to develop our understanding of what factors influence the microbial communities that establish in the built environment, and what factors affect the rate at which those communities change. We know that from the microbial perspective, the built environment represents a unique ecosystem. It's dry, it's climate controlled, and it's also regularly inhabited by humans who carry a lot of microbes around on them and in them. In this study, we explored 4 specific features that we hypothesized might affect the microbial communities that establish in the built environment and their rate of change. These features were the climate, the occupancy, so who was there and how many people were there, the surface material, and the location in the office or in the building. We achieved this by looking at 3 different cities with 3 different climates. So we looked at San Diego, Flagstaff, and Toronto. In each of the 3 cities, to explore the effect of occupancy, we looked at 3 different offices. And so we would pick 3 offices that had similar features in one specific city. And then in each of those offices, we would install 3 different sterilized surface materials, drywall, carpet tile, and ceiling tile in 3 different locations, on the floor, the ceiling, and the wall. Over the course of a year, we sampled each of these surfaces 78 times in 4 densely spaced sampling periods. So 1 during the summer, 1 during the fall, 1 during the winter, and 1 during the spring. First, the built environments that we inhabit are personalized in their composition. So while each of these materials was installed after being sterilized, over the course of this year, they've differentiated from each other. To the point that each of the microbial communities in each of these offices is unique. Next, the cities differ in the rate at which their microbial communities are changing. So what we've observed is that the communities in Flagstaff have changed a lot more rapidly than the communities in San Diego or Toronto. Next, and probably to be expected, the microbial communities on the floor are changing at a much more rapid rate than the microbial communities on the wall or the ceiling. And we think that this probably has to do with the fact that we're bringing dust into the office, for example, on our shoes, and we're kicking that dust around, and that is establishing much more quickly on the plates on the floor than on the ceiling and the wall. And finally, and mostly a technical point, one open question when we started this study was whether we would be collecting enough biomass to be able to accurately profile the bacterial communities. What we found so far is that this protocol does appear to work. So the protocol involves rubbing a dry swab over each of these materials and then sequencing the DNA that we obtain from that swab. And all of these results so far have been based on that swabbing technique and are giving us sufficient bacterial biomass to study those communities. If you're interested in this study and in learning more about the next steps, you should keep an eye on the microBEnet blog post. On the microBEnet blog, as I'm planning to publish all